Biotechnology. It's probably a word that many of you have heard, but maybe don't necessarily really know what it's all about. When we use biotechnology, we're using technology to manufacture or make products to improve quality of life. So we use biotechnology for all sorts of things in order to improve human life, animal life, or just any type of life in general. One of the things that we'll discuss when we talk about biotechnology is something called genetic engineering. Genetic engineering is when we make changes in the DNA code of living organisms using artificial methods. So in this case, we're talking about not through basic breeding. And in this case, we can change any living organism because we know all living organisms share DNA. We all use DNA for our genetic information. This is going to become very important when we talk about applications of genetic engineering in a minute. So one of the ways that we can carry out or study DNA in order to use it for genetic engineering is through a process called gel electrophoresis. And gel electrophoresis is used to separate and analyze DNA fragments or sections of DNA. Sometimes we don't want to look at all of the DNA because it's a really long code. We have 3 billion base pairs in the human DNA strand. We have over 30,000 genes. So sometimes we just want to look at a section. So the first thing that happens is we have several steps. We need to cut our DNA using something called restriction enzymes. So we have to extract the DNA by splitting the cells open. There are certain chemicals we can use in order to split those cells open. The second thing that happens is the DNA is separated using other types of chemicals. So we separate the DNA from the other cell parts. The third thing we do is we use these special enzymes called restriction enzymes to cut the DNA. And the idea is it cuts it at specific locations. And the way it does that is by recognizing specific base sequences. So for example, I might have a sequence of DNA, TAG, ATT, CTG, AAG, CCG, and then my opposing strand, or my complementary strand. And in this case, we have a special restriction enzyme called EcoR1, and it recognizes anywhere along the DNA strand the sequence CTT-AAG. And when it does, it makes a cut in this specific location after the second A in between the A and the G. So it's going to cut between the A and the G. When it does that, we end up with things called fragments or sections of DNA. And it turns out that some of these fragments have been cut unevenly. So we call them sticky ends because they're ready to bind to another segment of DNA. Once we've done that, we can take our DNA fragments after we've cut them with our enzymes, and we can put them in a solution with special things called probes. And these probes bind to the DNA, those sticky ends, in order to make them visible. The next thing, we take those fragments and we inject them into the wells of a porous gel. It's kind of like a really thick jello. And at the end are these little boxes or segments called wells. And you can use a special pipette to inject that solution of those DNA fragments into the wells. Once we've injected it, step 6, we can turn on an electrical current, and that gets applied to the gel. And the reason why we use the electrical current is because the DNA has a charge to it. So if I apply an electrical current, I know that my DNA is going to move towards the opposing charge. So in this case, the negative end is towards where the DNA was injected. The positive is at the other end. DNA is negatively charged when we cut it into fragments, so it's going to move towards the positive end. So it's going to move from the wells to the other end. So it's going to move based on size. And the reason why is this gel is kind of like a mesh strainer, kind of like you would strain spaghetti in when you cook spaghetti. The really big pieces of spaghetti don't go through and fall through the holes in the strainer. So they stay in the strainer, the small things go through, like the water. And in this case, the small fragments can fit through those holes and go farther along the gel. So the smallest fragments are going to be in the opposite end, and the large fragments are going to stay closer to the wells. So in the end, since we use these probes to make it visible, we can then eventually get it underneath an X-ray and have these probes come up and become visible to show our segments of DNA. Now right now it doesn't really mean much. We're going to talk about how to interpret the gel in a second. So what can we use it for, though? Well, we can use gel electrophoresis for lots of things. We can compare DNA genomes of different species. We can locate and identify different genes. Maybe I want to find the gene for hair color. Determine the parents of offspring or genetic relatives. We're going to talk about how that can be done in a second. Or matching DNA from a crime scene in suspects. We'll talk about that example too. The things you need to remember is that offspring inherit their DNA from each parent. So I get half of my DNA from my mother and half of my DNA from my father. And that's why we can do these types of things, like look for a parent of an offspring or compare DNA from crime scenes. The other thing you need to remember is that all organisms have DNA, and we use it for heredity. So I can look at the DNA of a frog and compare that to the DNA of a human because we all use DNA. It's a similar compound. So let's look at some of these uses in a little bit more detail. So let's look at comparing DNA from a crime scene. So a couple of things you need. You would need the crime scene DNA sample. You would need the suspect's DNA, maybe in some cases multiple suspects. And then you would take DNA from a random person that you know is not related to the crime scene. We'll talk about that in a second. So this would be my gel in the end after I ran that whole test. And on this side, we have things called standards. It's essentially our control. These are known base lengths to use as a comparison. And then I have C for my crime scene DNA, S for my suspect's DNA, and in this case, R for a random person DNA. So a couple of things to think about. If I know that these bands are segments of DNA of a specific length that got cut in specific sections by my enzymes, if the bands match up, it means that the DNA matches in terms of at least size, probably also the same sequence. So that means the DNA is a match. So in this case, does the suspect's DNA match the crime scene DNA? So here's my suspect and here's my crime scene. Do all of the bands match up completely? And in this case, they do. So since the bands match up, I have a match for my DNA to the crime scene, putting me to an idea that maybe my suspect matches the crime scene DNA and it at least puts them at the scene. Does it mean that they're a murderer or whatever it might be? Possibly not, but it shows that there was at least DNA there from that suspect. The fact that it matches is really one part of it. Taking the DNA from a random person helps to even further show that the odds of having a match are kind of slim. So maybe in some cases the odds of having a match might be 1 in 1 million. So if I take this random person and the random person does not match up with the crime scene, as it doesn't here, then it shows that you can't just take one random person's DNA and match it to the DNA of the crime scene. So it helps to further show me that this suspect is unique to this DNA that was found at the crime scene. Let's move on to a different example, paternity testing. So paternity testing is done when we want to compare the DNA from a child to a known parent and then hopefully compare it to a parent in question. So sometimes if you aren't sure of who the father of a child, you can compare the DNA of the child, compare the DNA to the mother, and then compare it to the DNA of the suspected father. Knowing that a child inherits half of their DNA from their mother and half of their DNA from their father, we should then be able to come to the conclusion that half of this child's DNA, or bands of DNA fragments, should match mom's, and half of them should match dad's. So if we look at this gel that we have here, we have our standards again for comparison. Mom, child, the suspected dad, and then a random person. Again, we're taking this random person in order to show that the child isn't just going to magically match up with a random person, to show that it's not by chance. So which bands match mom? So if I look at the child, I can see that these two bands match and these two bands match. So this child is the offspring of this mother. That's a good thing. Do any bands match the father? So here's dad. We have one match here, which is an opposite from the mother, and then another band here. So this child had 4 bands in this test. 2 of them matched moms, and 2 of them matched the suspected father. None of them matched up with our random person, which is good. It shows that they don't have a chance of being the father's child. So The nice thing is this child not only matched the mother, but also matched the suspected father. So it helps to show that hopefully this child has this father as their biological father, getting half of the DNA from dad and half of the DNA from mom.